So what is a gene? A gene is basically a section of DNA located at a particular position, the locus of a DNA molecule. A gene can code for amino acid sequence of a polypeptide and functional RNA, such as ribosomal RNA. The genetic code consists of a minimum of 3 bases that code for each amino acid, and this is because only 20 different amino acids regularly occur in proteins, so each amino acid must have its own code of bases on the DNA. Now, only 4 bases are present in DNA, A, T, C, and G. If each base coded for a different amino acid, that means that only 4 amino acids could be coded for. If only a pair of bases were used, only 16 different codes are possible, which is not enough as 20 amino acids occur. Therefore, 3 bases produce 64 different codes, which is more than enough. Here are some features of the genetic code. The genetic code is a degenerate code, meaning that most amino acids are coded for by more than one triplet. In simpler terms, one amino acid can be coded for by many different codons. A triplet is always read in one direction along the DNA strand, and the start of the DNA sequence that codes for a polypeptide is always the same triplet, methionine. If methionine does not form part of the final polypeptide, it is removed. 3 triplets do not code for any amino acid, and these triplets are called stop codes as they mark the end of a polypeptide chain. The genetic code is also non-overlapping, meaning each base is only read once. For example, it's read as 1, 2, 3, 4, 5, 6, 7, 8, 9, and not 1, 2, 3, 2, 3, 4, and 3, 4, 5. Lastly, the genetic code is universal, as each triplet codes for the same amino acids in all organisms, giving proof of evolution. Chromosomes are only visible when the cell is dividing, but usually are dispersed within the nucleus. When they are visible, they appear as 2 threads, joined at a single point by the centromere. And each thread is called a chromatid because the DNA has already replicated, meaning there are 2 identical DNA molecules. We already know that DNA is a double helix, and from the diagram here, we can see that this helix is wound around histones to fix it in position. This DNA histone complex is then coiled, and in turn is looped and further coiled before being packaged into the chromosome. If we follow the diagram carefully, we will see that a chromosome contains just a single molecule of DNA.
Structures of ribonucleic acid. Here we'll be discussing the RNA, its functions, and the different types of RNA. Firstly, RNA is a section of DNA that's transcribed into a single-strand molecule. RNA transfers DNA code from the nucleus to the cytoplasm and acts as a messenger. This is because RNA is small enough to exit the nucleus through the nuclear pores, unlike DNA. RNA also is made up of 3 bases, which are called codons, that code for one amino acid. Genome is a set of genes in a cell, including mitochondria and cytoplasm. And proteome, also known as complete proteome, is a full range of proteins produced by genomes. Now, an RNA is made up of pentose sugar ribose, adenine, guanine, cytosine, uracil, phosphate, and a phosphate group, as seen on the image. There are 2 types of RNA: mRNA, messenger RNA, or tRNA, transfer RNA. tRNA is a single strand folded into a clove leaf shape with one end extending beyond the other. This is the part of the tRNA molecule to which an amino acid can easily attach to. At the opposite end of the molecule is a sequence of 3 bases known as codon. Given that the genetic code is degenerate, there must be a tRNA molecule as there are codons. tRNA is specific to one amino acid and has an anticodon that's specific to that amino acid. mRNA's base sequence is determined by the sequence of bases on the DNA length through transcription. Here is a table comparing the structures and functions of DNA, mRNA, and tRNA. Transcription and splicing. Transcription is when a complementary section of a DNA sequence is made to form a molecule called pre-mRNA. Translation, which we will learn about later, is when mRNA is used as a template to which a complementary tRNA molecule is attached to, and the amino acid carry a link to form a polypeptide. The process of transcription is as follows. Firstly, the enzyme acts on a specific region of DNA, causing 2 strands of the DNA to separate and exposing the bases. The template strand, one of the 2 strands of DNA, pair up with the complementary nucleotides by using RNA polymerase and pre-mRNA is formed. Then a DNA strand rejoins as pre-mRNA is forming. Lastly, when the RNA polymerase reaches stop codon, it attaches and the production of pre-mRNA is complete. The next part of producing mRNA from DNA is splicing. Firstly, DNA is made up of exons, which code for protein, and introns that prevent protein synthesis as they don't code for anything. So in the process of splicing, base sequence corresponding to introns are removed and the functional exons are joined together. Polypeptide synthesis and translation. In this part of the chapter, we'll be learning about polypeptide synthesis. Here we have a tRNA. The T in tRNA stands for transfer, and a tRNA is responsible for the formation of amino acid and bringing them to the ribosomes to become proteins. A tRNA is able to produce amino acids by reading codons on mRNAs. Codon are 3 bases, and it's able to read these codons by having anticodons on the tRNA. Anticodons are complementary bases to the bases on the mRNA. And each tRNA is specific to a codon, so there are multiple tRNAs for each codon on an mRNA. Moreover, for example, the sequence of ACG means that the amino acid cysteine will attach to another end of that tRNA molecule. This anticodon will combine to the codon UGC on the mRNA molecule during the formation of a polypeptide. The mRNA codon UGC therefore translates into amino acid cysteine. The image below shows how a polypeptide is synthesized. First, a ribosome becomes attached to the starting codon AUG at one end of the mRNA molecule. The tRNA molecule with the complementary anticodon sequence AUC moves to the ribosome and pairs up with the codon on the mRNA. This tRNA carries a specific amino acid methionine. A tRNA molecule with the complementary anticodon UGC pairs with the next codon on the mRNA ACG. The tRNA molecule carries another amino acid, threonine. The ribosome moves along the mRNA, bringing together 2 tRNA molecules at any one time, each pairing up with the corresponding 2 codons on the mRNA. The 2 amino acids, methionine and threonine, on the tRNA are joined by a peptide bond using an enzyme, an ATP, which is hydrolyzed to provide the required energy. The ribosome moves onto the 3rd codon GAU in the sequence on the mRNA, thereby linking the amino acids threonine and aspartic acid on the 2nd and 3rd tRNA molecules. As this happens, the first tRNA is released from the amino acid methionine and is free to collect another amino acid methionine from the amino acid pool in the cell. This process continues in this way with up to 15 amino acids being added each second until a polypeptide chain is built. Up to 50 ribosomes can pass immediately behind the first so that many identical polypeptides can be assembled simultaneously. This synthesis of polypeptide continues until a ribosome reaches a stop codon. At this point, the ribosome and mRNA and the last tRNA molecule all separate and the polypeptide chain is complete. Here you can see a large-scale view of how polypeptide chains are formed by tRNAs. Lastly, how a protein is assembled. Sometimes a single polypeptide chain is a functional protein. Often a number of polypeptides are linked together to give a functional protein. Usually this is made up of the following steps. A polypeptide is coiled or folded, producing a secondary structure. The secondary structure is folded, producing a tertiary structure, and different peptide chains along with non-protein groups are linked to form a quaternary structure.